At Indiana, I joined a small group of visionary scientists, mostly physicists and chemists, studying the reproductive process of the viruses that attack bacteria, bacteriophages, phages for short. The phage group was born when my PhD supervisor, the Italian-trained medic Salvador Luria, and his close friend, the German-born theoretical physicist Max Delbruck, teamed up with the American physical chemist Alfred Hershey. During World War II, both Luria and Delbruck were considered enemy aliens and thus ineligible to serve in the war effort of American science. Even though Luria, a Jew, had been forced to leave France for New York City, and Delbruck had fled Germany as an objector to Nazism. Thus excluded, they continued to work in their respective university labs. Luria at Indiana and Delbruck at Vanderbilt, and collaborated on phage experiments during successive summers at Cold Spring Harbor. In 1943, they joined forces with the brilliant but taciturn Hershey, then doing phage research of his own at Washington University in St. Louis. The phage group's program was based on its belief that phages, like all viruses, were in effect naked genes. This concept had first been proposed in 1922 by the imaginative American geneticist Herman J. Muller, who 3 years later demonstrated that X-rays cause mutations. His belated Nobel Prize came in 1946, just after he joined the faculty of Indiana University. It was his presence, in fact, that led me to Indiana. Having started his career under T.H. Morgan, Muller knew better than anyone else how genetics had evolved during the first half of the 20th century, and I was enthralled by his lectures during my first term. His work on fruit flies, Drosophila, however, seemed to me to belong more to the past than to the future, and I only briefly considered doing thesis research under his supervision. I opted instead for Luria's phages, an even speedier experimental subject than Drosophila. Genetic crosses of phages done one day could be analyzed the next. For my PhD thesis research, Luria had me follow in his footsteps by studying how X-rays killed phage particles. Initially, I had hoped to show that viral death was caused by damage to phage DNA. Reluctantly, however, I eventually had to concede that my experimental approach could never give unambiguous answers at the chemical level. I could draw only biological conclusions. Even though phages were indeed effectively naked genes, I realized that the deep answers the phage group was seeking could be arrived at only through advanced chemistry. DNA somehow had to transcend its status as an acronym. It had to be understood as a molecular structure in all its chemical detail. Upon finishing my thesis, I saw no alternative but to move to a lab where I could study DNA chemistry. Unfortunately, however, knowing almost no pure chemistry, I would have been out of my depth in any lab attempting difficult experiments in organic or physical chemistry. I therefore took a postdoctoral fellowship in the Copenhagen lab of the biochemist Herman Kalker in the fall of 1950. He was studying the synthesis of the small molecules that make up DNA. But I figured out quickly that his biochemical approach would never lead to an understanding of the essence of the gene. Every day spent in his lab would be one more day's delay in learning how DNA carried genetic information. My Copenhagen year nonetheless ended productively. To escape the cold Danish spring, I went to the zoological station at Naples during April and May. During my last week there, I attended a small conference on X-ray diffraction methods for determining the 3D structures of molecules. X-ray diffraction is a way of studying the atomic structure of any molecule that can be crystallized. The crystal is bombarded with X-rays which bounce off its atoms and are scattered. The scatter pattern gives information about the structure of the molecule, but taken alone is not enough to solve the structure. The additional information needed is the phase assignment, which deals with the wave properties of the molecule. Solving the phase problem was not easy, and at that time only the most audacious scientists were willing to take it on. Most of the successes of the diffraction method had been achieved with relatively simple molecules. My expectations for the conference were low. I believed that a three-dimensional understanding of protein structure, or for that matter of DNA, was more than a decade away. Disappointing earlier X-ray photos suggested that DNA was particularly unlikely to yield up its secrets via the X-ray approach. These results were not surprising, since the exact sequences of DNA were expected to differ from one individual molecule to another. The resulting irregularity of surface configurations would understandably prevent the long, thin DNA chains from lying neatly side by side in the regular repeating patterns required for X-ray analysis to be successful. It was therefore a surprise and a delight to hear the last-minute talk on DNA by a 34-year-old Englishman named Maurice Wilkins from the Biophysics Lab of King's College, London. Wilkins was a physicist who during the war had worked on the Manhattan Project. For him, as for many of the other scientists involved, the actual deployment of the bomb on Hiroshima and Nagasaki, supposedly the culmination of all their work, was profoundly disillusioning. He considered forsaking science altogether to become a painter in Paris. But biology intervened. He too had read Schrödinger's book and was now tackling DNA with X-ray diffraction. He displayed a photograph of an X-ray diffraction pattern he had recently obtained, and its many precise reflections indicated a highly regular crystalline packing. DNA, one had to conclude, must have a regular structure, the elucidation of which might well reveal the nature of the gene. Instantly, I saw myself moving to London to help Wilkins find the structure. My attempt to converse with him after his talk, however, went nowhere. All I got from my efforts was a declaration of his conviction that much hard work lay ahead. While I was hitting consecutive dead ends, back in America, the world's preeminent chemist, Caltech's Linus Pauling, announced a major triumph. He had found the exact arrangement in which chains of amino acids, called polypeptides, fold up in proteins, and called his structure the alpha helix. That it was Pauling who made this breakthrough was no surprise. He was a scientific superstar. His book, The Nature of the Chemical Bond, essentially laid the foundation of modern chemistry. And for chemists of the day, it was the Bible. Pauling had been a precocious child. When he was 9, his father, a druggist in Oregon, wrote to the Oregonian newspaper requesting suggestions of reading matter for his bookish son, adding that he had already read the Bible and Darwin's Origin of Species. But the early death of Pauling's father, which brought the family to financial ruin, makes it remarkable that the promising young man managed to get an